Hello friends, today we will be talking about T cells, and in the T cells part 1 I have discussed about the naive T cells, how they are differentiated and they are formed in thymus. So if you want to know more about naive T lymphocytes, please go to the first part of my video on T cells. I will be sharing the link too. So let's now talk about naive T lymphocytes, which are naive because they have never come in contact with the specific antigen. That means they have not met their counterpart. Now, how the journey of these naive T cells takes place, let's talk about that. Naive T cells, which are in thymus, expresses L-selectin. L-selectin, which binds with high endothelial vanules, another expresses more high endothelial vanules expresses vascular adresin. So with this vascular adresin, L-selectin binds and this travels in blood streams, and they are then taken to peripheral lymphoid tissues. What are the peripheral lymphoid tissues? In next slide I will naming those tissues. Here I just want to talk about the travel of that is the journey of naive T cells. So this L-selectin, because of L-selectin and the vascular adresin, L-selectin present in naive T cells and vascular adresin present in HEVs, they bind and they travel in bloodstream. And another CC chemokine ligand, that is 21, CCL21, which is produced by HEVs and stromal cells in the peripheral tissues. They binds with the receptor which is present in naive T cells. And what is that receptor? This receptor is CCR7. So let's just revise this slide again. L-selectin present on naive T cells. This binds with vascular adresin, which is expressed by high endothelial vanules. Through this vanules, the naive T cells travel in thymus, from thymus to peripheral lymphoid tissues. And in peripheral lymphoid tissues, in the stromal cells of peripheral lymphoid tissues, and as well as HUVs, both produce CC chemokine ligand 21. This ligand 21 have an affinity to bind with the receptor which is also present on naive T cells. This is CCR7. So after this, what happens? Once this binding, this all kind of binding takes place between T cells, HUVs, and stromal cells of lymphoid tissues, the T cells are now recruited into peripheral lymphoid tissues. So what are the different peripheral lymphoid tissues? They are the lymph nodes, spleen, mucosal lymphoid tissue. These are all peripheral lymphoid tissues. So we were talking about journey of naive T cells. And if you want to know more about naive T cells, you can go back to my T cells part 1. So these naive T cells now come into contact with antigen presenting cells. When they have gone into the peripheral lymphoid tissue, there they come in contact with APCs, antigen presenting cells. And these antigen presenting cells carry peptides which are processed by them and which are in contact with MSC. Now you want to know more about MSC and how it is presented. I have a video on MSC also. Previous video is there. You can go back there. Here, let's talk about more about antigen presenting cells, because they are the ones which will be presenting antigens to the naive T cells. These first we will talk about dendritic cells. DCs are the full form of DCs are dendritic cells. They are the primary antigen presenting cells, and they are the ones which are responsible for activation of naive T cells. Two signals are also needed, but first these antigen presenting cells present the antigen. At mucosal sites, these dendritic cells form a dense network where they catch antigens, and this then they penetrate the surface. These DCs take up antigen by they are non-specific. Macropinocytosis takes place, or what is macropinocytosis? That is, they internalize the soluble antigens and then they find out the peptides, antigen peptides, which they present to MHC. and which are then taken to naive T cells. When either the micropinocytosis takes place, or this DC surface receptors also catches the antigen, and they are then taken to, with MSC, they are taken to naive T cells. Here we are discussing more about antigen presenting cell. One is B cell. B cell also takes up antigen by specific binding to cell surface immunoglobulin, while dendritic cells are not non-specific. But these B cells have specific bindings with immunoglobulins, surface immunoglobulins, mostly IgM, and then they take to T cells. And how T cells gets activated, we will be talking in the next slide. One more antigen presenting cell which I want to discuss is the macrophage, which engulf large particles through phagocytosis, and then it processes the peptides, and then it takes them to the surface and present to it to T cells. So these antigen presenting cells we have discussed. Some of antigen presenting cells, that is dendritic cells, which are more in number. Then B cells, mast cells are also one of the antigen presenting cells, while macrophages are also antigen presenting cells. Now let's I have put one slide on the role of antigen presenting cells, while I have discussed this in the previous slide also, but here I want to give you notes also. So I keep writing things so that you can have notes as well as my talk on it. So antigen presenting cells are breaking down antigens into peptide fragments in endocytic vesicles. That means in the vesicles, these are broken up, and the peptides which have to be presented, because specific antigens have to be presented to T cells. When T cells come in contact with these antigens which are presented by antigen presenting cells, then only these naive T cells become activated. So these are the antigen presenting cells present the antigen, and where peptides are loaded onto class 2 MSC proteins. Class 2 MSC proteins are also very much needed. Now what is class 2 MSC proteins or class 1? If you want to know more about this, go to my previous video, and I will be putting the link in this video also on MSC. So role of MSC should be known to everyone to understand these T cells activation. Then the act of processing antigen leads to what? Maturation of immature forms to mature antigen presenting cells. And once they become mature, then they do not leave mucosal surface, and they all migrate to lymphoid tissue. So antigen presenting cells also get mature once they present antigen to naive T cells. And once the antigen is presented to naive T cells, the journey here, the naive T cells will not be called now naive, because they have come in contact with the specific antigen. So for naive T cells to get activated, what two things are needed? First is antigen presenting cells which bring antigen, and with MSC molecule, class 2 molecule should also be there. And the second signal which they need is that the CD28, that is cluster of differentiation 28, present on T cells should also be there, which should bind with antigen presenting cells which are expressing B7 ligand. So antigen presenting cells also express, they have a specific antigen peptide fragment with them, and they have to express B7 ligands also. These B7 ligands are B71 and B72, which binds to CD80 and CD86, and interaction of CD40 ligand with CD40. So cluster of differentiation are present on T cells, and B7 ligand is essential for activation of T lymphocytes. With the B7 ligand will be expressed by antigen presenting cells, while this cluster of differentiations will be present on T cells. So this signal is very much needed. If no specific antigen is there, then if CD4 positive cells, I have told you what is positive selection. It is also in the first part of my T cells. If you want to know what is CD4 and positive selection, please go to that video also. Here we will talk that the CD4 cells do not come in contact with any specific antigen. That means they have not come into contact with the specific antigen. Then this CD4 cells passes back into the bloodstream and continues recirculating until it comes in contact with this antigen. So this naive T cell is basically a parent cell, and if it comes in contact, it's a cell, if it comes in contact with this specific antigen, that is specific peptide which can fit into this T cell receptor, then only this naive T cells will be getting activated. But for this activation, they need the antigen presenting cells with the specific receptor plus one signal, that is specific receptor TCR, that is T cell receptor, should fix in the specific antigen, as well as antigen presenting cells should express B7, and this B7 should bind with CD28. So two things are very much needed for B cell activation, that is B7, which is expressed by antigen presenting cells, as well as TCR receptor should get activated with the specific antigen brought by antigen presenting cells. So when they come into when CD4 comes in contact with specific antigen, it migrates no further, it stops there. So activation of T cells, now the journey of T cells have ended and activation of T cell have taken place. That was because of B7 CD28 interaction, which leads, already I have told you, it leads to the activation of T cell. One more thing happens here, that is CTLA4 is also produced by B7 binding. B7 also produce, once the B7 and CD28 get loaded together, they bind together, this complex also instructs the production of CTL4. What is the CTL4? It is the competitor of CD28. So CTL4 also then binds to B7. New naive T cells which are coming, which are specific for this T cell receptors, are specific for this specific antigen, are coming, but this activation has to be stopped also. You know that up regulation and down regulation both are very important. The T cell activation have to be stopped also at certain point. So who's going to down regulate it? That is B7 and CD28 competitor. What is CD28 competitor? The name of CD28 competitor is CTLA4. CTLA4 also binds with B7, and once CTLA4 binds to B7, the activation of T cells gets stopped, because CD28 is now B7 is not free to react with CD28. So this leads to down regulation. Now let's come back to like activation of T cells. T cells are activated when two signals are needed. First is TCR receptor should receive the specific antigen from antigen presenting cells, as well as antigen presenting cells are expressing B7. Now this B7 binds to CD28. Once this total two signal system has started, T cell undergoes G1, growth phase of cell cycle, and it begins to produce interleukin 2. Role of interleukin 2 is also very important. Interleukin 2 is essential, and it has the autocrine growth factor. It's the autocrine growth factor for T lymphocytes. Once interleukin 2 stimulates the T cell to undergo the process of cell cycle, the clonal expansion, that means T cells get clonally expanded, same copies of T cells start forming, and thus giving rise to effector cells which have a specific activity against a specific antigen, and thus T cell mediated immunity starts. And this is all about naive T cells journey to T cell activation, and how proliferation of T cells takes place, and how they are clonally selected. Third part is also there on T cells. We will be discussing in next lectures. Thank you.